Hello, this is Gina Piscatelli with the last lecture on the digestive system for Anatomy and Physiology 2. In this lecture, I'm going to review nutrient digestion and absorption in each part of the alimentary canal. I know you've got a lot of details, and so we're going to try to summarize it, including where does mechanical digestion occur? Where is each type of nutrient chemically digested? Those nutrients would be carbohydrates, proteins, and lipids. And where is each type of nutrient absorbed? That would include carbs, proteins, lipids, and then I would add water to that. And finally, how is food moved throughout each part of the digestive tract? So we're going to begin with the oral cavity. And recall that in the oral cavity, mechanical digestion occurs by mastication, which is chewing, and tongue movements. There's also chemical digestion of what we eat, specifically carbohydrates, because the saliva contains an enzyme called salivary amylase. The saliva also contains lingual lipase, which would break down lipids, but it isn't activated in the oral cavity. It's activated in the small intestine. So these processes of chemical and mechanical digestion produce what is known as a food bolus. There's no absorption of nutrients, and this food bolus is propelled by swallowing, and the term for swallowing is deglutition. And in the oral cavity, the first phase of deglutition occurs, the buccal phase, which involves tongue movements, and it's voluntary. Now moving on to the esophagus, there is no digestion, no mechanical digestion, no chemical digestion, and there's also no absorption. But of course, the food bolus is propelled through esophageal motility. And so we have the second phase of deglutition occurring. This is involuntary. It's called the pharyngeal esophageal phase. And it's mediated by the enteric nervous system. In addition, peristalsis moves the bolus from the superior esophagus towards the stomach, where at the very end of the esophagus, the cardiac sphincter relaxes to allow the bolus to enter the stomach. Now, in the stomach, we have this food bolus that is mechanically digested by a type of modified peristalsis that starts in the cardiac region and then continues to the pyloric region. There's also a little bit of erratic contraction which churns and mixes the bolus with gastric secretions. There's gastric juice. This process forms chyme. And now onto the chemical digestion. That gastric juice contains hydrochloric acid, gastric pepsin, and gastric lipase. And so what we get from that gastric juice is chemical digestion of proteins and some lipids. Also, lingual lipase is activated in the stomach, further enhancing lipid chemical digestion. Very little absorption of anything happens across the wall of the stomach. The wall of the stomach, the enterocytes, some of them produce intrinsic factor present in gastric juice, and that helps the stomach absorb vitamin B12, but mainly it's water and alcohol that's absorbed. Then for propelling the chyme that has formed from the stomach into the small intestine, we see this modified peristalsis, a wave of contraction that goes towards the pyloric sphincter, which is normally open. And so the chyme will squirt into the small intestine, but the pyloric sphincter will close, limiting how much chyme will enter the duodenum at any one time. And that is so the duodenum can neutralize the acidic chyme. So now the chyme has entered the small intestine, and as you know, the first part of the small intestine is the duodenum. The middle part is the jejunum, and the last part is the ileum. And in the small intestine, there is some mechanical digestion that takes place as segmentation occurs and mixes the contents in the small intestine with mucus, pancreatic juice, and bile. All of these, mucus, bile, and pancreatic juice, help to neutralize chyme. Remember in particular that bile, that they contain bicarbonate ions. Chemical digestion also occurs in the small intestine of all the nutrient types, carbohydrates, proteins, and lipids. That's because largely pancreatic juice and bile are emptied into the duodenum, but also the small intestine contains enzymes on its brush border. So the brush border enzyme enteropeptidase will help break down proteins. Pancreatic juice has trypsin, chymotrypsin, carboxypeptidase, which all break down proteins. Pancreatic amylase breaks down carbohydrates, and pancreatic lipase breaks down lipids. But the big chemical digester of lipids is bile, and it's really not an enzyme, but it emulsifies triglycerides to the extent that they get broken up. So it's not actually an enzyme that catalyzes a chemical reaction, but I put it under chemical digestion because of how it works. And then also in the small intestine, not only do we have the major site for chemical digestion, but we also have the major site for absorption. And the small intestine's histology reveals that it's an important site for absorption because the mucosa and submucosa form circular folds and villi and microvilli, which all enhance the surface area so that nutrients can be absorbed across that surface eventually into either the circulatory system or the lymphatic system. And we know that the monosaccharides and amino acids that are absorbed across the wall of the small intestine have to use transport proteins, but then they end up diffusing into the bloodstream and use the hepatic portal circulation to get to the liver first. And then eventually they will travel in the inferior vena cava back to the heart. The chylomicrons that are produced due to bile emulsification of fats, they will be released from the small intestine and enter lacteals, and those lacteals will carry the triglycerides that are in the chylomicrons to the circulatory system eventually. And for propulsion of the contents of the small intestine, we also, in addition to segmentation, see peristalsis or peristaltic waves that move the chyme and digested material throughout the length of the small intestine. And so finally, what's left over after absorption reaches the large intestine. And in the large intestine, we have mechanical digestion called haustral contractions where those pocket-like sacs contract periodically and in different regions, kind of like segmentation in the small intestine. We only have chemical digestion because of the presence of enteric bacteria. They're able to break down some of the indigestible, like cellulose, carbohydrate, fiber, maybe a little bit of lipid. But we don't really absorb the nutrients, so really it's just the bacteria breaking it down. It helps them produce ATP so that they can make vitamins K and B. And what we do absorb across the wall of the intestine in terms of nutrients would just be those vitamins, some ions or electrolytes, and water. So this is where the feces forms due to compaction of the contents, which occurs because water is being reabsorbed. To move the feces along the length of the large intestine, there is a type of motility or propulsion called mass movements. And those mass movements are able to move the contents of the large intestine a pretty good distance, centimeters, versus the haustral contractions, which only move it, to the neighboring segment or haustra. And to eliminate those feces, a mass movement sends feces to the rectum. And when the rectum distends, there's an involuntary reflex which causes relaxation of the internal anal sphincter. But as you know, we also have voluntary control over when we defecate, typically anyway. And so the external anal sphincter also needs to be relaxed, and that will only happen because a signal is initiated in the cerebrum, travels down the spinal cord, and to the skeletal muscle of that external anal sphincter. So I hope this review helped put a few things in perspective and kind of look at the big picture without being caught up in the small details. And thanks very much for listening.